Hello everyone, how are you all? Today we are going to learn about the seed development. Seed development mainly consists of three phases, that is embryo development, endosperm development and seed coat development. In this lecture we are going to learn about the endosperm development. I will explain about the embryo development and seed coat development in the next lectures. What is endosperm and what is its function? Endosperm mainly it provide food reserves to the developing embryo during seed development and during seed germination. Then how this endosperm used to form? During fertilization process, when two sperm nuclei released from the pollen tube, then one sperm nuclei fuses with the egg which will form the zygote, and second sperm nuclei fuses with the two polar nuclei which are located in the center of the embryo sac. Here in this picture you can see, then that sperm nuclei fuses with this polar nuclei and it forms the endosperm, that is primary endosperm nuclei. And this primary endosperm nuclei is the product of double fertilization and it is diploid in nature, because polar nuclei are already diploid in nature, where sperm nuclei is a haploid in nature, and totally it will become the triploid in nature. Its main function is to provide the storage reserves or nutritive material to the developing embryo during seed development and germination. And this endosperm is generally distinct in case of both angiosperms and gymnosperms, whereas in gymnosperms this endosperm used to be haploid in nature because there is a absence of triple fusion in case of gymnosperms, whereas in angiosperms we can observe this triple fusion. If you will take the families Orchidaceae and Podostemonaceae, the primary endosperm nuclei it will disintegrate. So endosperm development is completely suppressed in these two families, Orchidaceae and Podostemonaceae. That's why orchids are the non-endosperm seeds. And there are different type of endosperm development we can come across different species. So first one is the nuclear endosperm. Nuclear endosperm means here the primary endosperm nuclei undergo repeated mitotic divisions but without any cell wall formation. Here you can see there is no cell wall formation, but at the later stages we can observe the cell wall formation in the nuclear endosperm. Example for the nuclear endosperm is the coconut water. Second one is the cellular endosperm. In case of cellular endosperm, the nuclear divisions are followed by cell wall formation. Here in this picture you can see these mitotic divisions of the primary endosperm nucleus, it is followed by the cell wall formation. Here you can see. And third one is the halobial endosperm. Halobial endosperm means it is the intermediate between the nuclear and cellular endosperm. That means initially nuclear divisions takes place followed by cell wall formation. Here you can see half of the primary endosperm nuclei is showing the cell wall formation and half of the primary endosperm nuclei is not showing any cell wall formation. That's why it is intermediate between the nuclear and cellular endosperm. Examples for the halobial endosperm where we can observe are monocotyledons generally, like paddy, coconut and halobials. We can observe this type of endosperm. And another word is ruminant endosperm. In some cases we can see ruminant endosperm. This ruminant endosperm means endosperm with any degree of some irregularities or unevenness in its surface texture, then that endosperm we used to call as a ruminant endosperm. Why this ruminant endosperm used to form means because of unequal radial elongations during the seed coat development results in the formation of ruminant endosperm. This ruminant endosperm we can observe in Myristica fragrans and andrographies. And let us see some of the examples of these endospermic and non-endospermic seeds. Generally, endosperm reaches its maximum size at the stage of maturity in case of monocots, whereas in case of dicots this endosperm is completely utilized during the embryo development. So its development is not that much conspicuous in case of dicots. We may see it as remains as a small part of the seed, example groundnut and soybean. And the seeds which are having endosperm are generally known as endospermic or albuminous, while those with small amounts of endosperm are called non-endospermic or exalbuminous. Some of The examples for this endospermic and non-endospermic seeds are endospermic seeds, monocots we can found in rice and wheat, whereas dicots, castor and opium poppy we can see the endosperm. And in case of legumes, fenugreek, in fenugreek endosperm is there, whereas non-endospermic seeds we can observe in case of monocots, orchids where the endosperm development is completely suppressed, that is orchidaceae members, and water plantain, here you can see the seeds of water plantain. And in case of dicots, grams, that is Bengal gram, and peas and beans where the endosperm used to be completely utilized by the embryo. And sometimes we can come across some words like perisperm. What is perisperm means? In case of coffee and pigweed, generally endosperm used to be absent. In such cases, the perisperm act as a storage tissue. So this perisperm is generally diploid in nature and it used to be derived from the maternal nucleus. And coming to the another word, aileron layer. This aileron layers generally we can observe in monocots only, because in cereals and generally what will happen in most of the cereals and endospermic legumes, most of the majority of the cells in the endosperm are non-living. However, these peripheral layers of the endosperm, like if it is endosperm, this surrounding layers, some of the cells used to be living in nature. That living cells are generally called aileron layer. So what is the purpose of this aileron layer in case of monocots means? These aileron layers are generally filled with protein granules, and these proteins are most of the enzymatic in nature. Why they have to be enzymatic in nature means, during the seed germination, these enzymes helps in breakdown of the storage food materials and helps in release of that food material and provide to the this developing that is germinating seed, because during seed germination it requires lot of food material. That food material has to be break with the help of enzymes. That enzymes are provided by this aileron layer. That is the importance of this aileron layer. And let us see some of the different type of endosperms we can come across different species. Like how these endospermic cells look like means, they are isodiametric in nature and polyploid. Here you can see in this picture how these endospermic cells look like. And proteinaceous endosperm we can observe in most of the cereals, that is aileron layer itself. And starchy endosperm means that is cereals only we can observe. And oil endosperm we can observe in castor and coconut, because we used to extract from that oil endosperm only we will extract the oil. And cellulosic endosperm we can observe in ivory palm, that is hard endosperm. And hemicellulosic endosperm we can see in date palm. In date palm we can observe the hemicellulosic endosperm. We have to remember these examples: cellulosic endosperm, hemicellulosic endosperm. And seed with double endosperm, that is coconut, we can observe liquid endosperm, that is Nucellar endosperm and cellular endosperm. And stony endosperm we can see in betel nut, that is areca nut. These are the examples of different type of endosperms where we can come across different species. This is about the today's lecture. Thank you for your keen interest in this video, and remember to like and subscribe for more videos. If you like my videos and subscribe to my channel, it will give some encouragement to us to upload the more videos. And I wish you great success, health, prosperity and happiness. And if you will find this video is more useful, please share with your friends also. Thank you one and all.